The overall goal of this procedure is to identify protein interaction partners for a given protein of interest. This is accomplished by first growing the cells in cell culture media containing arginine and lysine labeled with different stable isotopes of carbon and nitrogen. The second step is to transfect plasmids encoding a protein of interest or a control plasmid into the labeled cells. Next, the cells are lysed and samples are immunoprecipitated from the lysates. Then, equal volumes of control and sample immunoprecipitates are combined and submitted for LC-MS-MS analysis. The final step is to analyze the mass spectrometry data to identify proteins interacting with the protein of interest. Ultimately, stable isotope labeling of amino acids in cell culture, or SILAC-immunoprecipitations, are capable of identifying large numbers of both direct and indirect interactions with a protein of interest from tissue culture cells.
The main advantage of this technique over existing methods such as tap tagging is that it removes the need for the investigator to send away individual bands for mass spectrometry. This removes an important source of bias whilst increasing sensitivity. Though this method can be used to provide insight into cellular protein-protein interactions, it can also be applied to other systems, such as studying pathogen-host interactions.
To begin, scrape SILAC-labeled HEK293 T cells expressing the GFP-tagged